Welcome to this episode. Today we're diving into a fascinating genetic question. Could the 8 survivors of Noah's Ark, as described in biblical texts, have been the genetic source of all modern ABO and RH blood types? It's a theoretical exploration grounded in population genetics and Mendelian inheritance, not a historical or theological claim.
Yeah, it's an intriguing intersection of genetics and tradition. So starting with the basics, the biblical account names 8 survivors: Noah, his wife, their 3 sons, Shem, Ham, and Japheth, and their wives. That's 8 individuals total.
Right, and from a genetics perspective, each person carries 2 copies of every autosomal gene. So collectively, that's 16 copies per gene. That's actually a decent amount of genetic material to work with, especially for something like blood type alleles.
Exactly. When we look at the ABO blood group system, it's controlled by a single gene with 3 common alleles, A, B, and O. These combine to produce the 4 main blood types, A, B, and O.
And, importantly, only those 3 alleles are needed to explain all ABO blood types. So, if among those 8 survivors all 3 alleles were present, it's entirely plausible that their descendants could produce every ABO blood type over a few generations.
Yeah, through standard Mendelian inheritance and assortative mating, the combination.
Now moving on to the RH factor, which is often simplified into 2 alleles, D, which is dominant and corresponds to RH positive, and D, which is recessive and corresponds to RH negative.
So, genotypes DD and DD result in RH positive blood, while DD results in RH negative. If even a few of the survivors carried the recessive D allele, RH negative individuals could appear naturally when 2 carriers have children.
Right, and the classic example is a DD times DD mating, which yields 25% DD offspring. Those are RH negative. This shows that RH negative blood types don't require new mutations to arise, just standard Mendelian segregation.
That's a great point. And since the ABO and RH genes assort independently, once all the necessary alleles are present, all 8 common blood types, A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, and O negative, can arise naturally over generations.
So, to generate this full spectrum, only 5 key alleles are needed: A, B, O for ABO, and D for RH. Given 16 copies per gene from 8 individuals, it's genetically plausible that all these alleles were represented in the founding population.
It's interesting how small that genetic toolkit is, isn't it? Just 5 alleles across 2 gene systems can explain all the common blood types we see today.
Absolutely. This ties into the concept of the founder effect in population genetics, where a small founding population gives rise to a much larger one. We see this in island populations or isolated communities.
Though founder populations often lose genetic diversity over time, initially they can carry enough allelic variation to produce diverse phenotypes. So if we accept the biblical narrative for the sake of argument, those 8 survivors would represent such a founder population.
From a genetic standpoint, that's plausible for the ABO and RH systems. But it's important to clarify what science can and cannot say here.
Right, science confirms that ABO and RH inheritance follow well-established Mendelian genetics. It supports the plausibility that 8 founders could carry the alleles needed for all modern blood types.
But science cannot verify the historical existence of Noah or the Ark survivors, nor can it determine their actual genotypes or confirm the occurrence of the flood. Those questions lie outside genetics and into history, archaeology, and theology.
So to sum up, from a modern genetics perspective, there's no inherent contradiction between the biblical account of 8 survivors and the later appearance of all 8 common ABO and RH blood types.
Provided the alleles A, B, O, D, and D were present among those survivors, standard inheritance mechanisms suffice to explain the observed blood type diversity.
This analysis clarifies genetic plausibility, but doesn't serve as historical verification. It's a neat example of how population genetics and Mendelian principles can inform discussions that intersect science and tradition.
Exactly. It's a reminder that genetics can illuminate possibilities without necessarily settling historical debates.
Well said. Thanks for joining us in this exploration.
Thanks. Until next time.